Hello, I'm Sally Kornbluth, president of MIT, and I'm thrilled to welcome you to this MIT community podcast, Curiosity Unbounded. One of the great pleasures of my job is the opportunity to talk with members of our faculty who recently earned tenure. Like their colleagues in every field here, they're pushing the boundaries of knowledge and possibility. Their passion and brilliance, their boundless curiosity offer a wonderful glimpse of the future of MIT. And this podcast is a way to share that inspiration with the world. Today, my guest is Giovanni Traverso. Gio has a foot in two worlds. He's an associate professor of mechanical engineering at MIT, and he's a gastroenterologist at Boston's Brigham and Women's Hospital. His work focuses on innovative methods for drug delivery, diagnostics, and biological sensing. So, Gio, welcome to the show.
Thank you. Thank you for having me.
Very happy to have you here. You moved around quite a bit when you were young, from England to Peru to Canada, then back to England. Can you tell us a little bit about these early moves, your early life, and how this all led you to MIT?
Absolutely. So I was born in England. My dad is from Peru and my mother's from Nicaragua, and they met in the UK. My dad had been sent to England to study economics, and then my mom was learning English. And, they met, and that's where they had me. That's where I was born. And then we left when I was just about to turn 5, and we moved to Peru. And I lived there for just under 10 years. So I spent the better part of sort of my younger years there. And then we emigrated to Canada. And we emigrated because of the instability in the country. And my parents really restarted their lives again and, found that Canada was very receptive really to hosting foreigners. And they built a new life there. And I went to school there. I spent 6 years there. And then when my time came up for thinking about university, I had started and been exposed to science and had an opportunity to do research. And as part of that, I started to explore different opportunities. And I applied back to the UK, but also in Canada. But I had a wonderful opportunity to go back to England to study. And specifically, one of the areas of interest was medicine. And outside of North America and most countries in the world, most folks will start studying medicine right after high school. So I went directly into medicine and I moved to Cambridge, England, and studied there at the University of Cambridge.
I did physiology part 1B and part 2 genetics at Cambridge.
So I was a part 2 genetics student too.
Oh, interesting.
Yeah. So I did my part 1 in medicine, and so I did physiology also, but part 2 was in genetics, yeah.
For our American listeners, this means in England, you really only do one single subject for the whole final undergraduate year. And it's very, very different from an American education where you're doing so many different subjects. And I have to say, I was kind of tripped up by the exam system in England because in America, you're doing continuous assessment where you're always taking exams all the time, as our students at MIT do. In England, they save one exam till the end of the year, and that's not easy for an American person to adapt to.
No, absolutely. I would say it took me a couple of years to get used to the system because it is sort of 100% finals across all the subjects. And
the temptation is to do nothing.
Exactly. And there are many distractions,
Exactly.
Yes,
exactly.
Yeah, absolutely.
I understand that you had an early co-op working in a lab. And how did you start getting interested in research? How did this sort of foster your love of research?
So in high school in Canada and Ontario at the time, the system essentially involved 13 years or grades. And so by grade 12, I had completed almost all of my requirements. And I had a wonderful chemistry teacher who really suggested I think about doing research in the lab. And she connected me with someone. That someone's name is Steve Shearer. He's now runs essentially all of the research at the Hospital for Sick Kids in Toronto, but at the time was a graduate student in the lab of Lab Chi Choi. Lab Chi Choi is known for essentially the linkage analysis and really identification of the gene for cystic fibrosis together with Francis Collins's group. And I worked in Lab Chi's lab as a high school student together with Steve Shearer. To me, this was unbelievable that this was an actual job. This was sort of from the high school student perspective. And I really enjoyed it and really, I would say, steered me in this direction, to this day.
Very interesting. So you work on ingestible devices that have medical function known as electroceuticals. Can you explain a little bit about this? How do these work?
Absolutely. So we do many things across sort of a range of areas. As you mentioned earlier, I'm a gastroenterologist, so I tend to think about how we can interact and engage with our bodies and the bodies, obviously, of patients to really help them through the gastrointestinal tract. And one of the areas that we've started to really understand to a much greater degree is essentially our capacity to stimulate the GI tract to modulate different hormones. And so let me give you an example. What we've shown is that we have the ability to either stimulate hunger or stimulate a sense of fullness depending on where and how we're stimulating the GI tract. And so essentially, these are capsules that one would swallow, and they can stimulate either the stomach or the small intestine. And depending on the stimulation, whether it's an electrical stimulus or whether it's a mechanical stimulus, it will induce these different patterns.
So presumably you're stimulating some nerve that is then feeding back to what's going on in the central nervous system. And so how would you compare this to what's happening with the use of GLP-1 agonists, which clearly are affecting appetite? So there are neural gut pathways that must be modulating all this. So in a sense, you're activating this potentially similar pathways?
Exactly. Just as, for example, if someone has a big bowl of oatmeal and they feel full in the morning, that feeling of fullness is really transmitted through nerves directly into the brain to really signal essentially that feeling of fullness or of satiety. And so what we're able to do, and this is part of what we're doing right now, is characterizing how that signaling is taking place and how we can encapsulate or capture that signal inside of an ingestible form factor to really facilitate patients to have control essentially over their nutrition sort of needs and modulate their own intake. And I would say similar to the effects of many of the drugs, including GLP-1 receptor agonists, as well as others that start to modulate that sensation or the need or drive for intake.
That's interesting. And presumably this could be done with fewer side effects than, the one thing about the GLP-1 drugs is they seem to be now having just a huge array of effects that are at least heretofore inexplicable. So presumably they're doing lots of other things, either directly on organs or in the brain, which hopefully these sort of things would be more precisely stimulating the gut brain. I don't know, the gut brain axis. Hard to know.
I think that's our hypothesis also, is that by really tuning and working with essentially the endogenous systems, that we're maximizing our own capacity to modulate intake, and rather than add an exogenous substance as such. That being said, I also want to recognize that GLP-1 receptor agonists are transforming sort of how we can look after patients. So there was just another paper in the New England Journal that I just saw looking at how these drugs are helping people, for example, with sleep apnea. But we've seen
it's kind of crazy.
Yeah, it's a really exciting time right now.
So what other type of health issues do your devices address? What other disease areas are you working in?
So we have a lot of development on really the core challenges of taking medication. And several years ago, one of the things that we started with the Gates Foundation involved addressing a fundamental aspect of essentially interacting with medication. And that is, when medications are administered on a frequent basis, in general, if a patient has an option of taking something more infrequently, they are likely to continue to take that medication. So this is the adherence or the compliance problem, although, I think adherence is a better term.
So like HIV regimens are notorious.
HIV, exactly. And the condition may, for example, affect the ability for the individual to take.
Yes.
So neuropsychiatric conditions, for example.
Yes.
We've done some incredible work, again, starting about 10 years ago, also together with collaborators, and now through a company that we started a few years ago, where, for example, we've been able to take a drug that's used for schizophrenia, where a patient would have had to take it once or twice a day, but now they just take it once a week. And that can really give a stable dose to help maximize essentially the care of that person. So that's one example, but there's also examples exactly like you said in HIV, but also in substance abuse disorders, where again, there's essentially the condition itself may impact the likelihood that the individual will continue to adhere. So again, it's more at the core of engaging with a therapeutic that exists and is known to have a positive impact. But the challenge that we're addressing is actually more of a behavioral one at the core of being a patient and engaging with that therapeutic.
To give our listeners a visual, I recall seeing one of your devices that you swallow this pill and then it opens up in your stomach almost like a spider, adheres to the wall, and then gradually releases drug.
That's exactly right. Yeah. It opens up, and because of the shape, because of the size and some of the mechanical properties, it can safely stay in the stomach, and we can control exactly for how long it stays. And using those systems, we are applying it across a range of different areas. But schizophrenia is, I would say, the one that's most advanced and currently in phase 3 clinical trials.
That's really interesting. You're doing something in diabetes as well, I believe. Tell me a little bit about that.
Yeah, so we have several things. So we've been collaborating with Novo Nordisk now for almost a decade, and we've worked with them in many different areas. So we started this collaboration back in 2015, really focused on developing systems that could enable the delivery of drugs like GLP-1 receptor agonists or insulin, where the capsule could achieve levels comparable to a standard injection. And so the technical term here is a bioavailability, and essentially having bioavailabilities that are greater than 10%. The systems that are on the market, for example, and folks may be familiar with a drug called Rysbilsis, which is semaglutide in an oral format, that has an oral bioavailability of about 1% to 2%. The systems that we're looking at are 10, 50, and greater. So they actually achieve comparable levels to the injection. And we've actually demonstrated that. We've developed a whole array of systems that enable that, some that look like a little leopard tortoise and they can self-orient, but certainly a whole array of systems. So that was sort of the initial body of work. But using those systems, you can deliver several drugs like GLP-1 receptor agonists, where now you can deliver a much lower amount than what is being included in the current capsules. So that starts to potentially address some of the challenges with the availability of these medications. That has been a major issue here in the US over the past few years. Now, about 3 years ago, we continued and really expanded that collaboration and have been working on cell therapies and really thinking about how we can develop curative interventions for patients, suffering from diabetes. And that certainly is a very active area of research, really thinking about everything from the manufacturing to the administration to how do these systems sort of survive in the body,
right,
and how do we monitor those?
Right. To have to not immunosuppress people, basically.
Exactly. That's exactly right. So those are very active areas of research.
Oh, very interesting. So you're an associate professor in MECE at MIT. You're a gastroenterologist at Brigham and Women's. This is a really perfect combination of skills for your work, but how do you manage to juggle both? Do you still see patients? And how does your clinical work inform how you're thinking about your laboratory work?
Absolutely. And I should add, I have 3 kids and I'm married. And so, there's a lot. But I think it's a team effort, right? And I think whether it's in the lab, whether it's in the clinic or at home, what we do is a team sport. And I think we have a wonderful team in all of these different venues. And so I cut down my clinical time. I used to have a regular clinic endoscopy session. But once I started at MIT, I really removed my outpatient practice or stopped my outpatient practice. And so now I've transitioned to only doing inpatient time in between the semesters. So usually in January is when I'm on service, and I do that at the Brigham. And I do that as part of a team. And so I work with fellows and trainees and also medical students, which I think is honestly a privilege to be able to interact with the trainees and, of course, the patients. But I think it gives me a unique perspective, and it's a very different, I would say, interaction than the one, for example, that I would have in class, like later today. When I'm seeing someone at the bedside, it's a very different interaction, and gives me at least a different perspective and different level of motivation for everything that we do.
Right. It's interesting. As a basic science, it always struck me that the clinical scientists who also had laboratories could actually see coming down the road clinical needs that would really help direct where you were going with the basic science, where it's not that it's not fundamentally curiosity-driven research. It's just that it also has a trajectory up front of a problem you would like to solve.
Absolutely. And I think our goal is to overall essentially have impact and really develop systems that will translate to patients, right? And so I would say, I love to read, and certainly several of our scientists and students are investigating fundamental questions. But I would say, have a tremendous amount of interest in ensuring that what we're doing and spending our time on will translate to help patients in the end.
Absolutely. Also, you mentioned your family life, your clinical work, your laboratory work, but you also hold 45 patents and have founded or co-founded a dozen companies that use your work. So how do you think about this? Is the entrepreneurial spirit something where did you get that from? And do you aim to instill that in your students?
To me, this is part of an arc where we start with a lot of the basic discoveries, developments, and that arc continues beyond the academic realm. And in order to have those technologies essentially help people in the end, beyond just the publication and beyond sort of the educational value, it involves basically generating products. And that often involves a startup or partnering or working and collaborating with other companies. And, I was first exposed to this as a graduate student with Bert Vogelstein at Johns Hopkins. And Bert, has spent his career as a cancer biologist, but also as someone who's very focused on translating the work from his lab and the lab of Ken Kinsler, his close collaborator, for many decades. And one of the things that I did during my PhD was actually work very closely with a small company at that time called Exact Sciences. And it was really through those interactions that I would say my initial curiosity was really stimulated, because I could see how the work that I was doing in the lab could eventually make it into a product. And eventually that work contributed to a test that is now on the market. I mean, it took many years, but that sort of really gave me a window. And then fast forward, I would say about 7-8 years, I was a resident at the Brigham, and I was looking into my fellowship or specialty training in gastroenterology, a big component of which is actually the research aspect. And I had reached out to Bob Langer here at MIT. And I remember meeting with Bob, and I said, look, I don't know anything about chemical engineering, material science. These are some of the areas that I'm interested in really learning about. And he's like, just come. And he was extremely welcoming. And, and to this day, I mean, he remains a mentor, collaborator. And that experience gave me a window and an opportunity to learn more about that translational aspect with respect to the intellectual property aspects, the considerations that go into new company formation. And, it's something that I try and pass on to the next generation. I do it through the classes that I teach. I do it in lab. And I continue to learn myself, from both the people that I work with as well as the students.
Yeah, it's actually so woven into the fabric at MIT in a way that I haven't seen a lot elsewhere. And, Bob Langer is sort of the epitome of this, right? And also your story of coming and saying, I don't know much about this, but I want to do this. This echoes in my mind, Bob's tales of his early career of people being willing to take a chance on him. And we all know how that turned out very well. You obviously don't have a lot of free time, given everything we followed. But, when you do have free time, what do you like to do?
I love to spend time with my family. My kids are 8, 11, and 13.
Wow.
And, they all play sports.
Okay, that's your weekend's done then.
Well, absolutely. Yeah. And so they all have tournaments every weekend or multiple games. But, I think it's wonderful to see them grow and develop their own interests. And, I love to be outside. I love to go biking. We live in the Northeast, so I do love, and I've tried to support winter sports also with our kids. So I'm a snowboarder. All of our kids either ski or snowboard. So we try, as soon as the snow comes, we do try and get out.
Oh, that's great.
Enjoy
that's
the
great.
outdoors, yeah.
The snow has been a little bit limited in the line. We could have a whole conversation about climate change. In any event, one final question about the work, which is, we think about drug delivery and bio-inspiration, and you mentioned cell and gene therapies. So how do you think about bio-inspiration in your work? In other words, how do you think about the intrinsic ways biological systems operate and how you leverage them for therapeutic end in your own work?
Great question. We have this large grant that we received from the government earlier this year, and we just gave our 6-month report. And as part of that, I had convened an external advisory panel. And one of the members had asked me about bioinspiration because they could see that in several programs. And I would say we look at bioinspiration in generally 2 different ways. One is essentially when we map out a set of challenges for a project, we do look for adjacencies in nature to see, has nature solved this challenge in a similar way? And that certainly does happen. And we have 2 projects right now. There's one that essentially looks at the evolutionary elements that underpin the behavior of a specific fish. And we've taken that learning and applied it now to a whole new family of devices. So that certainly is something that we do. And we collaborated in that specific body of work with a scientist over at Boston College. But we also look at bioinspiration to help convey our work. And so sometimes the complexity that is involved in developing these systems I recognize can be complex, but we also want to be able to share and essentially help the audience quickly appreciate sort of the nuances of the work. And so we also look at bioinspiration as a way of explaining or helping explain the work. So I would say those are the two ways that bioinspiration really weaves into what we do and how we explain the work that we do.
I mean, it seems like there's an infinite array of possibilities with the approaches you take. So many disease states and indeed sort of health states that could be stimulated by the variety of devices. So we really look forward to seeing what happens in the coming years from having sat in discussions of your promotion case that everyone was very excited. So now I'm extremely excited to see what comes next. So thank you for having this conversation.
Well, thank you so much for having me.
Absolutely. And to our audience, thank you for listening to Curiosity Unbounded. I very much hope you'll join us again. I'm Sally Kornbluth. Stay curious.
Curiosity Unbounded is a production of MIT News and the Institute Office of Communications in partnership with the Office of the President. This episode was researched, written, and produced by Christine Daniloff and Alexandra Steed. Our sound engineer is Dave Lashansky. For show notes, transcripts, and other episodes, please visit news.mit.edu/podcasts/curiosity-unbounded. And find us on YouTube, Spotify, Apple, or wherever you get your podcasts. To learn about the latest developments and updates from MIT, please visit news.mit.edu. You can follow us on Facebook, Instagram, and LinkedIn at Curiosity Unbounded Podcast. Thank you for joining us today. We hope you'll tune in next time when Sally will be speaking with political scientist Ariel White. Ariel's work touches on voting, voter rights, the criminal legal system, and how ordinary people can make a difference. We hope you'll be there. And remember, stay curious.